Hey, welcome! We're about to jump into a world that's completely invisible to the naked eye. It's a world where the search for cures and the safety of our medicines are actually linked by the smallest signals imaginable. We're talking about precision on a microscopic scale, where one tiny flicker can literally be the difference between being healthy and being sick. So, what is the connection? How does fighting something like cancer relate to making sure a new medicine is safe? Well, it all boils down to one thing: finding these unbelievably tiny signals in a huge ocean of what scientists call biological noise. All that random background chatter happening inside our bodies all the time. All right, let's get into it. Okay, first up, let's talk about our body's invisible switches. See, deep down inside every one of our cells, these microscopic signals are basically running the show. They're the conductors of our own internal orchestra, controlling, well, pretty much everything. At the very heart of this whole control system is a process with a mouthful of a name: protein phosphorylation. But don't let the name scare you. Just think of it like a tiny biological on/off switch. When a little phosphate group gets added to a protein, click, it flips the switch. That tells the cell what to do next, whether that's to grow, divide, or even to self-destruct. It's like a traffic light system for our entire body. But what happens when those switches start to go haywire? When the signals get stuck on or maybe stuck on off? Well, that's a recipe for disaster. This is directly tied to some of the biggest diseases we face today: cancer, neurodegenerative disorders like Alzheimer's, and metabolic syndromes. So finding these broken switches, that's a huge mission for modern medical research. So how in the world do you see something that small? That brings us to some incredible technology. It's called microliquid chromatography inductively coupled plasma mass spectroscopy. Yeah, I know. Let's just call it MU-LC-ICP-MS. It sounds super complex, but for researchers, this thing is an absolute game changer. Let's quickly break down how this amazing machine actually works. First, scientists take a sample and separate all the proteins. Then, they use enzymes to kind of chop up those proteins into smaller bits called peptides. And now, here comes the magic part. The machine scans for two key elements: phosphorus and sulfur. That's what tells them which switches are flipped on. And finally, they can measure exactly how strong that on signal is. It's how we make the invisible visible. Okay, so we can see the signals for disease now. That's huge. But what about the medicines we use to fight those diseases? This brings us to the other side of the precision coin: finding hidden dangers in our medications. This is all about making sure they're manufactured with incredible purity. You see, when companies make a lot of modern medicines, especially protein-based ones, they use living cells as these tiny little factories. Think of cells from hamsters or even E. coli. But after the medicine is made and purified, some leftover genetic debris from those factory cells can get left behind. We call this residual host cell DNA, or rDNA for short. So why do we even care about this leftover debris? Well, it can pose some really serious risks. It could potentially contain viral genes that might become active, or it could have what are called oncogenes, genes that might promote cancer. And on top of that, it can even trigger an unwanted immune response in the patient. Finding and getting rid of this stuff is absolutely critical for safety. So that leads us to the solution. We need a kind of genetic safety net. How do we find these tiny, hidden dangers to make sure every single dose of medicine is as safe as it can possibly be? The gold standard method here is something called qPCR. The easiest way to think about it is like a genetic photocopier, but with a built-in counter. It can find one specific little piece of that residual DNA, make millions and millions of copies of it, and as it does, it creates this fluorescent signal. By measuring that glow, scientists can figure out the exact amount of that genetic debris that's there, right down to the tiniest little fragment. So how good is qPCR compared to older methods? Well, it's not even close. It's a clear winner. For starters, it's 10 times more sensitive. And get this, it gives you results in about an hour instead of 5 or 6 hours. Plus, it's incredibly specific. It's that combination of speed and precision that makes it the undisputed industry standard. And just how precise do we need to be? Global regulators like the FDA have set the limit at no more than 10 nanograms of this stuff per dose. To give you some perspective, that's like trying to find one single grain of sand in an Olympic-sized swimming pool. That's the level of precision we're talking about to keep everybody safe. So being able to achieve that level of precision is just incredible. But it does bring up a pretty big question. If these tools are so powerful, both for research and for safety, why isn't every single lab out there using them? Well, the answer really lies in what you could call the precision bottleneck. It's this set of very real, very big challenges that just come with the territory when you're doing this kind of advanced science. Getting your hands on this kind of technology is not simple. For one, it requires super specialized and very expensive equipment. Then, even if you have the machine, you need these complex data analysis systems to make any sense of the mountains of information it spits out. And on top of all that, running a lot of tests and keeping up with all the strict regulations, it just adds more and more layers of difficulty. For a lot of labs, those barriers are just too high to overcome on their own. So, how do we break through this bottleneck? Well, that brings us to the final piece of the puzzle here, this idea of science as a service. It's a whole model designed to make this level of precision accessible to anyone who needs it. And this isn't just some theory, it's actually having a huge impact in the real world. Just take this quote from Sarah Thompson over at UC Berkeley. She said, "Their thorough analysis enabled me to pinpoint specific interactions that I hadn't considered before, which significantly improved the direction of my research." That shows you right there how getting access to this kind of expertise can just accelerate discovery. And here's another one from a senior research scientist, Dr. Emily Carter. She said the results were invaluable because they helped her find key markers linked to brain function. This really shows how these services are helping scientists from all different fields connect the dots in these super complex systems. So this collaborative model is really the key to breaking that precision bottleneck. Researchers get to use cutting-edge tech without the massive price tag. They get experts who can help them turn all that raw data into real, actual insights. And they don't have to worry about meeting all those tough regulatory standards. The bottom line, it speeds everything up, from finding new disease markers to developing life-saving drugs. And all this brings us to one final thought. As the science we use gets more and more complex, as we try to understand these incredibly complicated diseases and create new cures, is this kind of collaboration, this teamwork, is that the next big frontier for discovery? It's a huge question, and the answer could really shape the entire future of medicine. Thanks so much for tuning in.